a core problem for many of us in origin of life science is figuring out what were the first entities capable of displaying key lifelike properties such as self-propagation and adaptive evolution, but are also simple enough that we can imagine them spontaneously arising in the absence of a prior adaptive process. So there are many models out there, but the one that we tend to rally behind is this notion that the first entities capable of self-propagation were cooperating sets of chemical species that were spatially localized on a mineral surface and could grow laterally autocatalytically using fluxes of food and energy present in the environment. And in addition to being able to self-propagate, it turns out that these slimes, as we like to refer to them for surface-limited metabolisms, could also be evolvable if rare side reactions either alter or expand the existing catalytic core and add new reaction modules, or cores as they're referred to. And in the context of a natural environment where we have mineral surfaces bathed in, for example, seawater that's rich in organics and other compounds building up by various prebiotic synthesis pathways, we imagine that the burial and continual exposure of new mineral surface would tend to enrich for systems that are better at getting from grain to grain, or at least can self-propagate faster. So the goal of our research is to test the slime model for the origin of life by essentially recreating that little seascape I just showed you and asking if we can enrich for these systems using a protocol that we've developed called chemical ecosystem selection. And the protocol is very simple. It essentially involves combining prebiotic soups containing many dissolved organics and other compounds in the presence of mineral grains, allowing for putative slimes to emerge. And in this figure, I've depicted several kinds of slimes that maybe differ in their colonizing abilities, denoted by different colors. And at the end of an incubation period, we transfer a small subset of colonized grains to new reaction vessels containing fresh compounds, so fresh food and fresh uncolonized mineral surface. And we do this over many generations with the hopes that we would enrich for systems that are better at getting from grain to grain. Now, the nice thing about this framework is that it's chemically agnostic, meaning that you can test many different scenarios based on your preference. But for the purposes of this talk, I'm going to give you an example of a recipe we've been using extensively and have been getting some cool results with. So the soup that we use is what we call an enriched miliary soup. So it's actually a synthetic soup that we build, that we make using off-the-shelf reagents. And we make a number of additions to reflect, for example, what you might find in an ocean, so salts, transition metals. And then we also add potential sources of chemical energy. And the one I'm going to highlight here is ATP. But I'm also going to say and acknowledge that ATP is probably not a very prebiotically plausible phosphate source, but we used it anyway, so I'm going to show you that data. As for the mineral, we've tried a few, but the one, again, I'm going to focus on is pyrite, this iron sulfide mineral, which we grind up into fine powders and then use in our experiments. And we combine it with our soup in these little reaction vessels, which are sealed serum vials. We flush the headspace with nitrogen, and then we also autoclave them at the end of each generation just to ensure sterility. So that's the recipe. And of course, being able to deploy this protocol implies that we'd be able to detect slimes if they were to emerge. And the way we do that is by assuming that if slimes did emerge and change over time, we'd be able to see that reflected in changes in a sealed reaction vessel, either in the bulk solution or on the mineral surface. So that's what we look at. So here, what we have on the y-axis is the amount of free phosphate we detect in our solutions at the end of an incubation period. So if you remember, I mentioned we add ATP. So we can use simple colorimetric assays to measure the amount of free inorganic phosphate that's released from the hydrolysis of that ATP molecule and track how that changes over time as we keep selecting. So what we see on the y-axis are different lineages. So the gray bars are 10 independent lineages that were set up in the same way with the same ingredients, but propagated independently of each other. And that orange bar is the average of a control pool that's set up using the same reagents at the same time, but only exposed to one history of transfers. And what you can see from this plot, hopefully pretty clearly, is that as the number of generations increases, we have a progressive decline in the amount of phosphate we detect. And so we infer that to mean, pretty simply, that a history of transfer matters. And then another thing that we measure is basically how much light is absorbed by that bulk solution at the end of a generation as a proxy for how many light-absorbing compounds, including some of the organics that we have in our solution, remain after such incubations. And what we find is that vials or reaction vessels that have a long history of transfer, in this case 18, have significantly lower absorbance than our control set. And we infer that to mean that some of the light-absorbing compounds, including the organics, have been depleted from the solution in some way. So this coincident reduction in both the amount of free phosphate and light-absorbing compounds and organics in our bulk solution led us to inspect the surfaces of our pyrite grains to see if we could observe any systematic differences. And we used electron microscopy to do this. So here's a representative micrograph of grains taken from a control vial. So again, this was one that only had one transfer in its history. And this is what we see when we look at experimental grains. So this one had 18 rounds of transfer in its history. And what you can see, hopefully, yes, you can see them pretty clearly, are these really distinctive fractal structures. Now, to make a long story short about what our best guess as to what these things are is, so we don't actually think these are the putative slime or the phosphate or organic consuming structures. We think that they're salt crystals that are fouled with organics or growing on a layer of organics that are on the pyrite surface, and that gives them this distinctive fractal or seaweed dendrite shape. So with these results in mind, we decided to repeat the experiment. This time we carried it out for longer, and we also took measurements at the end of each generation this time. And this is what we saw in the free orthophosphate concentration. So this time I've pulled it to reflect the average of our 10 independent experimental lineages. And you can see pretty clearly this pretty distinctive oscillatory pattern where we have an initial, what we call our boom phase, that is this linear decline in the amount of free phosphate, and then a sudden reversal in that trend where we go back to our initial phosphate levels, if not overshooting it a little bit. So we have a few hypotheses to explain this behavior. One is that that initial decline in free phosphate, which is now a repeatable instance, although you'll notice the timing is a little bit different this time, and we think that that's because we use slightly different batches of our enriched miliary soup that might vary a little bit. We think that that's reflective of the self-propagating state taking hold and growing on the mineral surface. Now, the bust phase can be explained in a number of ways. The one that we like to consider is kind of an ecological perspective, where you essentially have resource depletion. This system gets to a state where it consumes its resources and then collapses, and the process starts all over again. I also want to point out that we've looked at the fractal coverage at different points here, and we have some qualitative arm-wavy estimates of how they correlate with the phosphate pattern. So it turns out that we get the highest incidence of fractals when the phosphate is also at its minimum. So we think that this provides circumstantial evidence for what we think these critters are. They're not actually the slimes, they are a proxy for them, essentially. But one thing, I mean, is pretty clear from this graph alone is that it seems like we've enriched for some kind of nonlinear chemical system, which is good news in that I view nonlinearity as being a prerequisite for self-propagation and evolution. So on that front, we're good. But I want to get at the source of this nonlinearity a little bit more specifically. And so what we did for that is we actually looked at what happened intragenerationally. So what happens when you set up your miliary soup with your pyrite, you autoclave it, and then you track how these different proxy traits change over time. And what we saw in the phosphate, at least, is this really, again, pronounced oscillatory pattern that looks to be damped. And notably, it also looks like there's a lot of potential for nonlinearity even after our typical transfer period, which is 2 days. I don't know if I mentioned that yet. We also looked at the total absorbance and how that changed over our time course. And you can see that it has its own dynamics. It doesn't necessarily completely overlap with what we see in the phosphate. But it does seem to indicate what we would expect to see with the fractals. So again, this is very qualitative. We don't really have a good way to quantify the extent of fractal coverage yet. But what it looks like is that the fractal frequency increases over the first 72 hours, which is coincident with this trough in the UV-Vis data, after which we no longer observe them, and they never seem to come back, as far as we can tell, at least over the period we looked at. So is it a slime? Well, we're not sure yet, but I do think that these results are really interesting, and I think they kind of give us an idea of what kinds of chemistry and what kinds of dynamics we can expect to find using this protocol. But what we would really need to convince ourselves that this has the potential to be a slime as we've defined it is to demonstrate that it can self-propagate. And although we think it's likely that there is a self-propagating stage, we would really need to demonstrate that the nonlinearity is actually confined to the mineral surface. And that's part of ongoing work that also includes clarifying the chemistry of the slime. I'm sure you're dying to know as much as I am what this thing looks like, what it's made up of chemically, and we just have no idea at this point. And we also, to that end, want to test complementary conditions and do things like swap out ATP for a more realistic source of phosphate. And finally, I just want to end and comment on this question of, is it evolvable? I think that's ultimately what we want to know and what would make us really excited to find. And so we're moving in that direction right now, and we're basically looking to see if we can find changes in the rate of propagation over time in response to our protocol. That figure down there is just hypothetical data, just to show you that we have some notion of what that should look like. Even if we have kind of different modes of evolution. And then finally, I think a long-term goal, or kind of an idealistic one, would be to create environment-specific strains, if you will, that are adapted to slightly different conditions like pH, temperature, dissolved oxygen, and have them be fitter or more efficient in their own respective environments. So with that, I'd just like to quickly acknowledge the long list of people that make this possible and that actually do these experiments. So of course, my PI, David Baum, our co-author and collaborator, Jim Cleaves, our funding sources that come out of the NASA NSF Ideas Lab. So I'd like to thank our collaborators as part of that. Of course, our amazing research group. And then finally, just a quick shout out to the artist of some of the illustrations that you saw in my talk that actually come from a comic we co-wrote about how you can use science to solve seemingly intractable questions like the origin of life. So thank you very much.
We have time for a couple of questions while our next speaker comes up here. Yeah, right there in the center.
Hello, thank you for your very interesting talk. I'm glad I got to hear it. So I'm very curious, you have these fractals that are interacting with the pyrite crystal, correct? And they go away with this, during the cycling. But I'm wondering if at the end of your experiment, if you look very carefully at the pyrite crystals, is there some evidence that that there was some interaction, that was there? Are they leaving a record of themselves, if you will?
So the short answer is we're not sure yet. So we're just getting in the realm of in-depth surface analysis. That's something that's been quite difficult to do using the grain format that we're using. So we just don't know. So it's possible that because we don't see them come back, that it's not that they're gone per se, but they might have diffused to the point where we don't actually see them kind of localized in these really cool fractal structures. So that's one possibility. But we just simply don't know at this point.
Well, was your control with no pyrite, or did you show that?
So in the selection experiments, we have controls that are set up strategically alongside the generations we measure at, so that they only have one generation in their history. So we'll compare samples that have identical conditions, except one set will have, say, 20 generations of transfers versus just one.
But you haven't done it with no pyrite?
We've done the time series experiments with no pyrite, and convinced ourselves that at least the intergenerational dynamics we see require the presence of pyrite. But we have not yet done selection experiments in the absence of pyrite, and we plan on doing that.
All right, one more over here.
Okay. So you saw fun dynamics within a single vial without transfers, right? So I was wondering, do you know if there's anything different that happens if you transfer out of that vial at different time points? Like, does it starve and stop being able to propagate, or does it propagate differently if you pull it out at different moments? Like, that might tell you if it's depending on non-equilibrium chemistry.
Yeah, that's a great question. The only real data that I have from experiments like the ones you've just suggested are very preliminary. We did them just once, actually in parallel with the time course I just showed you. And we found basically we did a 1-day transfer and a 2-day transfer. And we kept those lineages separately. So basically one had a 2-day generation time, the other had a 1-day generation time. And we did find that the boom-bust patterning that you got in those two where it was quite different, the timing was quite different, which suggests that when you do that initial transfer, where you are in that oscillation that you get intergenerationally matters, but that's all very preliminary. And we're going to do experiments much more extensively and try to use that intergenerational dynamic pattern to strategically select different generation times. But yeah, that's a great question. We're thinking along those lines very much so.
Okay.
And one more quick question in the center. Yeah.
Quick question. It seems to me that one of the sort of important assumptions that goes into this slime model is that you have like relatively slow chaos for these like organics onto surfaces, and it seems like that's actually a pretty, like at least compared to what you've done, like a pretty simple experiment that you could do to sort of limit or constrain what types of species you're interested in.
Yeah, yes, and I'm not an expert in nonlinear dynamics, so I don't know what to say to your point about.
Well, I guess have you guys tried doing the experiment? You take a single component, add it to pyrite, and just see what's the kinetics of it sticking onto pyrite.
We have not, but we plan on doing kind of reduction experiments where we leave out whole groups of compounds and maybe, I don't know, narrow it down to just a handful, but we haven't done that yet.
Thank you, Dr. Vincent.